Imagine designing a complex building, but you don't actually draw the floor plan.
Like you just leave it blank.
Yeah, exactly. You just build a bunch of, flexible blank rooms and let the tenants who move in completely rewire the electricity, move the walls, and just, redesign the entire ventilation system to suit whatever specific job they happen to be doing that day.
I mean, that would be an absolute nightmare for an architect.
Yeah.
But from a biological perspective, it's actually a masterpiece of efficiency.
Exactly. Because for a long time when we looked at the structural cells of our lymph nodes, we basically viewed them as, static architecture.
Right, like concrete.
Yeah, just the passive concrete and steel scaffolding where the real action, the immune response actually took place. But today we are doing a deep dive into this groundbreaking 2026 paper published in the journal Immunity.
And it is a fascinating paper.
It really is. We're looking at an incredible stack of data, including single-cell sequencing and spatial transcriptomics that just completely upends that whole static view.
Yeah, the central thesis of this research is that our immune cells actually force the structural cells of the lymph node to, remodel their environment on the fly.
Yeah.
It is a highly dynamic, bidirectional conversation.
Okay, so let's unpack this. Our mission today is to thoroughly understand how these structural cells, they're called fibroblastic reticular cells, or FRCs, how they build these custom training camps.
Yeah.
We're going to dig into a genetic mechanism called the Notch2 signaling pathway. And most importantly, we want to explore why this microscopic city planning is like the absolute deciding factor in whether or not your body remembers a past viral infection.
So let's start with the master builders themselves, the FRCs.
Yeah.
As structural cells, their primary job is to organize the secondary lymphoid organs, like your lymph nodes, into distinct zones.
Okay.
And they do this by secreting specific chemokines. It basically creates these chemical gradients that pull different immune cells into their designated areas.
Right, so B cells are drawn to the B zone and T cells are drawn to the T zone.
Exactly. And the traditional view is pretty straightforward, right? We just assumed the T zone was this one giant uniform waiting room for T cells and dendritic cells. But this paper's first major finding completely shatters that idea.
Oh, totally.
Using really advanced immunofluorescence microscopy, the researchers discovered that the T zone is highly compartmentalized. It's not just a single room. It operates much more like a dartboard.
I love that visual analogy, a dartboard.
Yeah. So picture a dartboard. The center of this base, the bullseye, is built by a highly specialized subset of structural cells that we call CCL19 high T-zone reticular cells.
Because they're pumping out massive quantities of that CCL19 chemokine.
Precisely. And crucially, these central cells express a specific surface protein called MADCLM1.
Okay, so that's the bullseye. And then surrounding that,
You have the outer ring. This area is built by structural cells that produce very little CCL19. And instead of MADCLM1, they express a completely different marker called gremlin-1.
Wait, gremlin-1?
Yeah, gremlin-1. So physically, these two networks of structural cells segregate perfectly into a central core and an outer shell.
Okay, wait, hold on. I have to push back on this purely from a, historical perspective.
Sure.
We have been looking at lymph nodes under microscopes for over a century. If there is a giant structural bullseye sitting right in the middle of the T-zone, how on earth did immunologists just miss it for decades?
It's a fantastic question. And it really comes down to a fundamental paradigm shift in how we interpret biological markers.
What do you mean?
Well, for years, MADTM1 was used almost exclusively to identify a different type of cell called marginal reticular cells. And those sit way out at the very edge of the lymph node.
Right, on the margins.
Exactly.
Yeah.
Because immunologists expected MADTM1 to only be at the edge, they often used it in flow cytometry to just isolate those edge-dwelling cells, completely ignoring the spatial context.
Ah, so they were blending up the tissue, pulling out the MADCM1 cells, and just assuming, oh, these came from the outer edge.
Yes, exactly. But this paper proves that approximately 80% of all MADCM1 positive structural cells in the lymph node are actually sitting right in the center of the T-zone.
Wow, 80%.
80%. It means that decades of older studies might need reinterpretation because scientists were, likely studying a mixed bag of cells dominated by this newly defined central core.
That is wild. It just shows how much context actually matters. So if the FRCs are the architects building this dartboard, what actually dictates the floor plan? Like what tells a structural cell to become a high-producing CCL19 bullseye cell instead of a gremlin-1 outer ring cell?
That leads us to the molecular zoning permit. The researchers ran single-cell RNA sequencing to look at the active genetic blueprints of these specific subsets.
Okay.
And when they isolated the central bullseye cells, they found a massive, undeniable signature for the Notch signaling pathway, specifically driven by the Notch2 receptor.
Okay, so Notch2 is the receptor sitting on the outside of these structural cells. When the researchers realized this, how did they prove that it was actually, controlling the architecture?
They went to mouse models. They engineered mice where they could specifically delete the Notch2 gene, but only in the structural cells of the lymph node.
That is incredibly precise.
It is, and the results were absolute. When they deleted Notch2, but not Notch1, by the way, it had to be Notch2, the central CCL19 high bullseye virtually disappeared.
It's like the city completely revoked the zoning permits for the downtown high-rises.
That's a great way to put it.
And what's fascinating is that the buildings themselves didn't vanish. I mean, the total number of structural cells stayed exactly the same, right? They just lost their identity.
Precisely. The cells physically changed, they increased in size, they got more granular, they stopped pumping out CCL19, and the entire area just reverted to the outer ring blueprint. They started expressing Gremlin-1 everywhere.
So the bullseye just melts into the outer ring.
Exactly. And to understand how a single receptor can cause such a massive identity shift, we really have to look at the mechanics of the canonical Notch pathway, because it is literally a mechanical marvel.
Right, because this isn't just a chemical cascade. There's actual physical movement happening here.
Yes. So when the Notch2 receptor is activated on the surface of the cell, it's physically pulled.
Wait, pulled? Like under tension?
Under tension. It gets pulled, which exposes a cleavage site. Then an enzyme comes along and literally snaps the receptor in half.
Oh, wow.
The inside portion, we call it the Notch2 intracellular domain, or N2ICD. It breaks off from the cell membrane, travels deep into the nucleus, pairs up with the transcription factor called RBPJK, and just begins physically remodeling the cell's DNA.
And the researchers proved this using ATAC-seq.
They did, yeah.
Which, just for context, ATAC-seq is a technique that measures which parts of the chromatin are physically unwound and open for business versus the parts that are tightly spooled up and closed off. What did the ATAC-seq data show when Notch2 was removed?
The numbers are just staggering. When Notch2 was lost, over 6,000 regions of chromatin newly opened up, and over 7,000 regions slammed shut.
7,000?
Yeah. The entire genetic landscape of the cell was rewritten.
Okay, let me pause right here and do a quick recap so we can make sure we're all picturing the architecture correctly before we look at what's actually living in these rooms.
Good idea.
So first, the lymph node T-zone isn't uniform. It has a central CCL19 high bullseye and a Gremlin-1 outer ring. Second, the Notch2 receptor acts as the master switch. When it gets activated, it physically snaps, dives into the nucleus, and completely rewrites thousands of lines of genetic code to maintain that bullseye identity. Do I have that right?
You have it exactly right. But what makes this truly remarkable is that this isn't just a developmental switch that gets flipped when you're an embryo.
Right.
Using a specialized mouse model, the researchers turned off Notch2 specifically in adult mice, and the bullseye still vanished.
So this active, massive DNA remodeling is happening continuously in your adult lymph nodes right now.
Every single day.
Which brings us to the most crucial question. If Notch2 is the receptor-like, the lock on the door, what is the key? And more importantly, who is holding it?
So receptors like Notch2 require a ligand to activate them. Through a series of blocking experiments using neutralizing antibodies, the researchers identified the key. It's a ligand called JAGED1.
JAGED1, and they found something incredible about the timing here, didn't they?
They really did. They discovered that blocking either Notch2 or JAGED1 for just 48 hours caused the central bullseye to collapse.
48 hours, that is so fast.
So fast. Within 2 days, the structural cells underwent transdifferentiation. They dynamically shifted their state back to the outer ring identity.
Okay, here is where it gets really interesting for me. Who is holding that JAGED1 key? You might assume it's just a signal from neighboring structural cells, or maybe blood vessels telling them how to grow.
Sure, that would make sense.
But it's not. The key is held by the immune cells themselves, specifically mature dendritic cells that express the CCR7 homing receptor.
Which flips the entire architectural paradigm right on its head. I like to think of these dendritic cells as demanding tenants.
Yes. I was hoping we'd get to this. Tell me about the demanding tenants.
So normally we think of immune cells passively wandering into a lymph node and just taking whatever seed is available. But these mature dendritic cells don't do that.
They don't just sit in the waiting room.
No, they arrive in the T-zone, grab the structural landlord by the collar using their jagged 1 ligand, engage the landlord's Notch2 receptor, and literally force the landlord to remodel the space specifically for them.
They demand the renovations.
Exactly. They demand that the structural cells start pumping out huge amounts of CCL19.
It's basically an aggressive cellular takeover. But to prove this wasn't just a correlation, the researchers needed to remove the tenants and see what happened to the building.
And they used a really elegant method to do this involving diphtheria toxin.
Right, so they engineered a mouse model where only the dendritic cells possessed a receptor for diphtheria toxin. It acts as a highly precise genetic scalpel.
So when they administer the toxin, it selectively wipes out just the dendritic cells, leaving every other cell of the lymph node completely untouched.
And what happened to the landlord when the demanding tenants were evicted?
Without the dendritic cells there to provide the jagged 1 signal, the central structural cells lost their CCL19 high identity. The bullseye just collapsed.
Wow.
And they went even further. They created a mouse where they deleted the jagged 1 gene only in dendritic cells. So the cells were still physically there, but they lost their key.
And?
Same result. The architectural bullseye vanished.
Okay, so to consolidate the mechanism here for the second wave of information, the Notch2 receptor on structural cells is unlocked by the jagged 1 ligand. Mature dendritic cells carry this ligand. And this relationship is totally bidirectional and continuous.
Yeah.
If the dendritic cells leave or stop signaling, the specialized central zone simply ceases to exist within 48 hours.
It's a continuous loop of instruction and adaptation.
But this begs a massive functional question, like why go through all this trouble? Why do the dendritic cells need to force the creation of this very specific CCL19 high central zone? What is the actual immunological consequence of this microscopic city planning for you, the person listening?
To answer that, we have to look at what that massive cloud of CCL19 chemokine actually does. It acts as a powerful, localized magnet.
Good.
There's a specific subset of dendritic cells called CDC1s. These cells are the absolute masters of cross-presentation. Their entire job is to take pieces of a virus or a tumor and present them directly to CD8 positive T cells.
The killer Ts.
The killer T cells, exactly.
Right, so the CDC1s migrate deep into the central zone. And because they force the structural cells to pump out CCL19, that gradient pulls the killer T cells directly into the center as well. It physically clusters them right next to the dendritic cells.
Exactly. It basically forces the teachers and the students into the same tiny, highly concentrated classroom. And physical proximity is everything in immunology. T-cell activation isn't just a brief handshake. It requires sustained signaling over hours, sometimes even days.
So what happens if they aren't forced into that classroom? The researchers ran an in vivo experiment mimicking a viral infection using an antigen called OVA combined with poly-IC. What happens when the bullseye is missing?
Well, in normal mice, the killer T cells expanded beautifully. They fought the mock infection. But in the mice missing Notch2 in their structural cells, the ones without the bullseye, the expansion of those killer T cells dropped by 40%.
40%. That is a massive hit to the immune response.
It is, but the sheer number of cells wasn't actually the most important defect. The real failure was in the type of memory being formed.
Right, because during an infection, your body creates a fork in the road for these T cells.
Precisely. Your body makes short-lived effector cells, or SLECs. These are the frontline infantry. They fight the immediate battle and then undergo apoptosis. They die off.
They do their job and leave.
Right. And the researchers found that SLECs form completely normally, even without the Notch2 bullseye.
But you don't just need infantry for today, you need guards for tomorrow.
And that's exactly where the system failed. The defect was specifically in the generation of memory precursor effector cells, or MPICs. These are the elite veteran cells that stay behind, surviving for years to protect you from ever getting sick from that same pathogen again.
So without the bullseye, they don't form.
Without that precise, Notch2-driven physical clustering, these memory precursor cells failed to develop properly. Furthermore, the memory cells that did manage to form were functionally stunted.
Stunted how?
They produced significantly less of the vital immune cytokines, like IFN, gamma, and IL-2, which are required to mount a robust defense later on.
So if we step back and look at the whole picture, this physical spatial clustering driven by the Notch2 pathway isn't just about organizing cellular traffic. It is biologically essential for writing long-term immune memory. If the cells don't sit in the exact right seat for the exact right amount of time, surrounded by that specific architectural niche, they simply do not learn how to remember the virus.
That is exactly the takeaway. The architecture itself provides critical survival and differentiation signals to the immune system.
Now, I have to ask the ultimate question here, because, I mean, this is a breathtaking display of genetic engineering and cell tracking in mouse biology. But does this actual mechanism happen inside human lymph nodes? Is this relevant to our own immune responses right now?
The researchers provided very compelling evidence that it absolutely does. They didn't just stop with the mouse models. They utilized human single-cell RNA sequencing datasets alongside a pretty cutting-edge technique called spatial transcriptomics.
For those who might not work with spatial transcriptomics every day, how does that differ from traditional sequencing?
So traditional sequencing requires you to dissolve the tissue to look at the RNA.
You essentially blend it up.
Right, you blend it up so you lose the map of where everything was. You know it's there, but not where it came from. Spatial transcriptomics allows you to take a microscopic slice of intact human tissue and map exactly where every single piece of RNA is physically located on the slide.
So you can literally see the architecture and the genetics at the exact same time. That's incredible. What did the human maps show?
They found the exact same dartboard blueprint in human lymph nodes. They observed a distinct central CCL19 high zone, surrounded by a GIR-1 high outer zone.
And what about the Notch2 pathway? Is that the same?
Completely conserved. The human structural cells in the central core strongly expressed NOTCH2, along with its active downstream target genes like HES1 and HES4.
And the dendritic cells?
Sitting right next to them were human CCR7 positive dendritic cells carrying the JG1 ligand.
So the demanding tenants are operating in humans as well. But, just because the cells are sitting next to each other doesn't inherently prove they are talking to each other. Did they analyze the actual communication networks?
We did, utilizing an algorithmic tool called CellChat.
CellChat?
Yeah, CellChat. This algorithm analyzes single-cell data by looking at the expression of receptors on one cell type and the expression of corresponding ligands on another. It then calculates the mathematical probability of physical interaction between those specific cell clusters.
It's basically a computational wiretap for cellular communication.
Pretty much. And what CellChat predicted was amazing.
What did it show?
The algorithm confirmed that the most significant, highly probable communication network occurring in that space was the JG1 NOTCH2 pathway, flowing directly from the mature dendritic cells to the CCL19 high structural cells.
Wow.
It is an evolutionarily conserved mechanism. Your lymph nodes are using the exact same zoning permits and demanding tenants as the mice.
That is just incredible. So what does this all mean for how we understand our own biology? It means your immune system is a highly adaptable, dynamic architect. Your structural cells are not passive concrete. They are in a continuous, active conversation with your immune cells. Your dendritic cells are constantly remodeling your lymph nodes via the JAG1 and Notch2 pathway, tearing down and renovating the training facilities based entirely on who is currently occupying them. And all of this is to ensure your body can remember and fight off the diseases you encounter.
And understanding this mechanism raises a really profound, forward-looking question for modern medicine.
Ooh, tell me.
We know that in severe diseases like cancer, tumors sometimes form their own immune structures. They're known as tertiary lymphoid structures, or TLS.
Right, these are basically rogue lymph nodes that form directly inside or adjacent to a tumor bed.
Exactly. And sometimes these structures help fight the tumor, and sometimes the tumor hijacks them to suppress the immune system. But now that we know the exact molecular zoning permit, the JAG1 Notch2 pathway that specifically builds CD8 positive memory hubs, it opens up a fascinating therapeutic frontier. Well, could we one day engineer therapies that force tumors to build these highly efficient training camps against themselves?
Oh, wow. That is a staggering thought. Like delivering a localized drug that tricks the tumor's own stroma into building a perfect CCL19 high bullseye, drawing in killer T cells to just eradicate the cancer from the inside out.
Exactly. It shifts our focus from just trying to stimulate the immune cells to actually engineering the battlefield itself to ensure they win.
It's an entirely new way to think about immunotherapy. We're going to leave you to mull over that incredible concept. Keep asking questions, keep looking past the obvious structural scaffolding in whatever field you study, and never forget that inside of you right now, there is a microscopic, dynamic city constantly rebuilding itself to keep you safe. Thanks for joining us on this deep dive.